So chances are you've heard about CRISPR, more formally known as CRISPR-Cas9, which has been described kind of broadly as this revolutionary gene editing tool that's taken the scientific world by storm. But what does that really mean? So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it's derived from bacterial immune systems to protect them from invading viruses. So during viral infection, a bacterium will send out a protein called the Cas9 protein, which is a nuclease, meaning it cuts DNA, to snip out part of the virus's DNA. The viral DNA sequence, which is called a spacer, is then inserted in between sequences of bacterial DNA. And those sequences are the CRISPR, the clustered repeats that are palindromic, meaning that their bases read the same forwards as backwards. So ATTA, for example. So now the viral DNA, the spacers, have been incorporated into the bacterium's genome so that if the virus attacks again, that bacterium can then recognize it and defend itself. So how this works is the viral DNA is transcribed into a special kind of RNA called CRISPR RNA, cRNA for short. And the cRNA combines with another special kind of RNA called trans-activating RNA, tracRNA for short, to form guide RNA, gRNA. So the gRNA binds to a Cas9 protein and it guides it to the viral DNA sequence that the cRNA is complementary to, it matches. There's also a recognition site on the target DNA called the PAM sequence, which is necessary for the Cas9 protein to cut it. So the PAM is a short DNA sequence in the CRISPR locus that comes right after the targeted DNA sequence. The PAM sequence is crucial in order to prevent the Cas9 from cutting the bacteria's DNA. Because remember, the target DNA sequence on the virus is identical to the one that's been embedded in between the bacterial DNA. So the bacterial CRISPR locus doesn't have the correct PAM sequence next to its spacer, so therefore the Cas enzyme can't target and destroy the bacterial DNA. So once the guide RNA takes the Cas9 protein to the target sequence, it searches for the PAM sequence. And then once the PAM sequence has been found, the Cas9 protein can then recognize and cut its target. But without that recognition site, it can't do that. And so once the match has been identified, the Cas9 protein then cuts the target DNA, inactivating the virus so it can't affect the bacteria. So in 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, figured out a way to turn this bacterial defense mechanism into a gene editing tool that could be used on human genomes as well as those of other organisms. They actually won a Nobel Prize for their work in 2020. So here's how their discovery works. Scientists identify a gene that they want to modify. And then they synthesize a guide RNA out of RNA nucleotides that are complementary to the target gene. So for example, if the target gene was ATTG, the guide RNA would be UAAC. And then that guide RNA then attaches to a Cas9 protein that they've gotten from bacteria, and it performs the process that I described above, guides it to the gene, checks the PAM sequence, cuts the DNA. This is the really important part. So after the Cas9 makes a cut, there's now a break in the DNA sequence, which is obviously bad. So the cell is going to want to repair that as soon as possible. Now, depending on what the desired outcome is, the repair process is going to happen in one of two ways. The first way is called non-homologous end joining, and this is used for gene knockout when you want to inactivate a gene. So repair enzymes are going to either put or remove random nucleotides, putting in small insertions or deletions, indels for short, at the break site. Now, this method is very error-prone, so chances are it's not going to be restored to the original nucleotide sequence. And now, these indels can cause frame shift mutations, which leads to the loss of gene function. So now the gene is knocked out. The second method is called homology-directed repair. Now, this is used for gene knock-ins, so inserting a new gene, mutation corrections, or just modifying an existing gene. So scientists provide a synthetic DNA template to the cell that's identical to the target gene, and it's identical to the original DNA sequence, except for the desired edit that they want to introduce, such as a different base pair to fix a mutation, or an entirely new gene. Now, the reason that this tool is so revolutionary is that no gene editing tool has ever allowed for such precise, cost-effective edits to DNA. And it's being used in our world right now to solve issues like genetic diseases and climate change. It's already been proven to be effective. For example, it successfully cured sickle cell disease, and it helped bring back the dire wolf, and this is just the beginning. However, some people both in and out of the scientific community are worried that this power might lead to abuse, as it already has been, and that we might overstep ethical boundaries with it, as is the case with any new scientific breakthrough that happens. We have to treat it with caution. CRISPR is still a fairly new technology, and it definitely has the potential to do a lot of good. So as research and experimentation continues, we'll see a lot more about what it can do and what it should and should not do. But for now, it's definitely one of the most exciting developments in science. So hopefully you find it as exciting as I do. Thanks for watching. Really quickly, I just wanted to include this diagram. I think it is a pretty good visual that explains how CRISPR works. So as you can see, here is the guide RNA. It's complementary to the target gene, and it's showing the Cas9 protein where to cut. This pink little globular structure is the Cas9 protein. You can see it's represented by the little scissors. It is literally a molecular scissor. It is about to cut into the target DNA sequence. And then here we have the PAM sequences, or the protospacer adjacent motifs, but PAM is much easier to say, helping the Cas9 protein identify where exactly to cut. So hopefully this diagram makes the CRISPR process much easier to visualize.